So next up, we have Priscilla Ambrosi, who is a PhD candidate in Talia Lerner's lab at Northwestern University, and will tell us about dopamine circuits driving the transition to habits. All yours. You have to unmute yourself.
Thank you. Okay. Hooray, Zoom. Hi, everyone. My name is Priscilla Ambrosi. Thank you for the introduction, Gunnar. Yeah, it's a pleasure to be here today. Okay, habits often develop from goal-directed behaviors. Right now we're all washing our hands to protect from coronavirus. But with repetition, you might develop a habit or a behavior that is driven by a stimulus response association. So every time you touch a doorknob, you wash your hands. And this is a benign habit, but habits can become maladaptive and turn into compulsions or obsessions. So if you constantly feel that your hands are covered in germs and you wash them to the point of damaging your skin, that is no longer beneficial. It is far removed from the goal of protecting you from infection. And I am interested in the circuits underlying this transition in the brain. Thankfully, we can model this transition in rodents by teaching them to nose poke or lever press for a reward. And with that, previous studies have shown that the basal ganglia seems to play an important role in habit formation. Specifically, the dorsal striatum, which can be subdivided into dorsomedial striatum or DMS and dorsolateral striatum or DLS. So if as animals learn a new motor task and transition from being goal-directed to habitual, there's a shift in synaptic activity from DMS to DLS. And if you lesion or inhibit DLS, you hinder habit formation. More specifically, if you lesion the dopamine cells that project to DLS, you also hinder habit formation. So together, these findings point to this putative circuit connecting DMS to DLS through substantia nigra as important for habit formation. And this circuit is part of a larger model called the ascending spiral. And the strongest evidence supporting the existence of this circuit comes from a tracing study in non-human primates. So here I'm showing you the striatum subdivided into caudate and putamen. And just for the sake of clarity, I'll be referring to these as the rodent homologue, DMS and DLS. And here we have substantia nigra. So what the study has shown is that there's this topographical organization of the projections from substantia nigra to striatum, such that medial goes to medial and lateral to lateral. And this organization is mostly conserved on the opposite direction, with the exception that DMS also projects to lateral substantia nigra. So if you put these two things together, there's this overlap between terminals from DMS and cell bodies from DLS projecting cells. And with this, it has been hypothesized that DMS activation can somehow excite DLS projecting dopamine neurons, leading to dopamine release in DLS. And the repeated activation of the circuit would then strengthen incoming synapses from sensory motor cortex, building those stimulus response associations required for habit formation. Now, there's a growing literature relying on this model as the mechanism behind habit formation, but there's a few important missing pieces here. So first, neuroterminals from DMS, axon terminals from DMS are GABAergic. So a direct connection between DMS and SNC would actually lead to the opposite effect on dopamine release. So the current ascending spiral model assumes that this connection between DMS and SNC is indirect, such that DMS activation inhibits tonically firing GABAergic neurons in SNR, thus disinhibiting DLS projecting dopamine neurons, allowing them to fire more and release more dopamine. Okay, but then if you are an electrophysiologist like me, you might be a bit skeptical about the anatomical data supporting this circuit, given that the overlap between axon terminals and cell bodies is not necessary nor sufficient for the existence of a functional circuit, especially not a polysynaptic circuit. And that's where I come in. So I am testing the existence of the circuit in mice. And here I just have a cartoon showing the striatum of mice. substantia nigra pars compacta, SNC, where dopamine cells are, and pars reticulata, SNR, where GABAergic cells are. And today I'll discuss two questions I'm trying to answer. One, is there a polysynaptic circuit of disinhibition from DMS to DLS projecting neurons? And two, does DMS activation actually lead to dopamine release in DLS? And to answer these questions, I'm using ex vivo electrophysiology and in vivo fiber photometry. All right, so for the first question, I'm also using projection labeling and optogenetics to study this long-range circuit in brain slices. And these techniques allow me to record specifically from these DLS projecting dopamine neurons while stimulating DMS-targeted GABAergic neurons in SNR. So I'm not interested in the general connection between SNR and SNC, but really the connection between these two subpopulations based on their inputs and outputs. Okay, so how do I test this circuit? First, I need to insert chondrodopsin in this particular subpopulation. How do I do that? I take an adult mouse, I inject a virus encoding Cre into DMS, and this particular virus has been shown to move transsynaptically in the anterograde direction, such that cells that are postsynaptic to DMS will also be infected and carry Cre. And that allows, so cells, in partly for this experiment, cells that are DMS targeted in SNR will carry Cre. Unfortunately, some dopamine cells will also carry Cre, given the direct connection between DMS and SNC that was previously shown by my advisor. So to prevent chondrodopsin expression in dopamine cells, I do two things. One, I use a Th2A FLIPO mouse in which FLIP is expressed in Th positive or dopaminergic cells. And two, I inject a virus encoding Creon flip-off chondrodopsin in substantia nigra. So this virus ensures that chondrodopsin is only expressed in cells that have Cre, but not FLIP. That is how I get chondrodopsin expression only in this subpopulation, cells that are Cre positive and therefore DMS targeted, but FLIP negative and therefore non-dopaminergic. To complete the setup, I also inject retrobeads in DLS, and these are fluorescently labeled latex beads that travel retrogradely from axon terminals to cell bodies. And this allows me to find DLS projecting cells in substantia nigra and PET from them while I'm stimulating this subpopulation of SNR cells. All right, and when I record from these DLS projecting dopamine neurons, I use a high chloride internal and a cocktail of drugs to isolate monosynaptic inhibitory synapses. And here is an example cell. You see the current in response to a light pulse, a 5 milliseconds light pulse. The thin traces are 10 individual sweeps and the thick trace is the average. And this cell is in fact inhibited by these subpopulation of SNR cells. If it wasn't, this would be a flat line. And here are the average responses from all of the cells I recorded. As you can see, there are some flat lines, but in fact, around 80% of the cells I recorded were inhibited. So that's really exciting because this is the first functional evidence of the existence of this synapse, which supports the existence of the first, of the whole polysynaptic circuit. All right, cool. So it looks like the circuit exists. But does DMS activation leads to dopamine release in DLS? To answer that, I injected a virus encoding D-light into DLS, implanted an optic fiber over DLS to record dopamine transients in that area. And then I injected another virus encoding crimson in DMS and implanted an optic fiber over substantia nigra so I can activate terminals from DMS in substantia nigra while the animal goes about his business in an open field box. All right, brace yourselves, here comes the data. It's a whole lot of nothing. So surprisingly, in the 4 animals I recorded, the activation of these DMS terminals in SNR does not seem to cause a dopamine transient in DLS. So, my first thought was great, my setup doesn't work. So to check that I could detect dopamine transients in DLS, I did a positive control in which I injected Cre-dependent crimson in the substantia nigra of that Cre mice. So now I have crimson expression in dopamine cells in SNC. So the key difference between these two is that here I am stimulating terminals from DMS, and here I'm stimulating cell bodies in SNC of dopamine cells. But in both, I am recording dopamine transients in DLS. And this was a good sanity check because I did find a nice dopamine transient in DLS after activating dopamine cells in SNC. So this is actually a bit confusing because the circuit seems to exist, but doesn't work the way we predicted. Why not? So one possibility is that the effect of this polysynaptic circuit is masked by the simultaneous activation of this direct connection between DMS and SNC, and these two pathways cancel each other out. Another possibility is that this polysynaptic circuit increases tonic firing of the dopamine cells, but doesn't cause them to burst. And it's unclear whether increased tonic firing translates to dopamine release in striatum. So maybe you need coincident excitation, or maybe even coincident modulation of release of the axon terminals in DLS to unmask the effects of this polysynaptic circuit. And if you do need any extra inputs encoding saliency or value, those would not be present in the open field experiment I performed. So I hope I have convinced you that there's a lot we don't know about this circuit. And before we fully rely on it as a mechanism behind habit formation, more experiments needs to be done. In fact, my ex vivo data is the first functional evidence supporting the existence of this circuit, but my in vivo data suggests that it doesn't work the way we predicted. So, I hope we don't go into a second lockdown so I can continue to do experiments and figure out what we're missing. And with that, I want to thank you all for watching. Thank you, Neuromatch organizers and all these beautiful people and funding sources.
Thank you so much, Priscilla. That was a very nice talk. Are there any questions by either the panelists or the audience? Please submit them. So when you showed the depiction of the pathways, it seemed like that there might also be a pathway going the other way. Is that just an optical illusion, or can you comment on that?
Oh yeah, you're absolutely right. So according to the anatomical data shown by Haber, the anatomical data suggests that this ascending spiral exists, but not a descending spiral going from DLS to substantia nigra to DMS. However, I have collected some preliminary data that, let me see if I can jump slides here. Yes, so I have collected some preliminary data showing that this descending spiral also seems to exist. So if you just invert the injections of the viruses here in the retrobeads and you do the same experiment, I also found, again, this is preliminary results, but it seems to be lower probability of connection, but it's there.
So is it just an impression, or does it target a different portion of the substantia nigra?
Oh no, that's not an impression. That is real. So this was previously shown by my advisor that these DMS projecting dopamine cells are more immediately located, and yeah, the DLS projecting dopamine cells are more laterally located. And you can also see that these DMS targeted versus DLS targeted SNR cells also have some topography in substantia nigra.
Right. So then if you have both circuits working together, how does that affect the interpretation of your in vivo results?
Good question, right? And if you consider the bridging collaterals, I mean, I don't know. It's complicated. I don't know. I just need to do more experiments to somehow, dissect all of these different pathways. The problem with basal ganglia is that it's one loop inside of other. It's just it's loopy. So that's actually a very hard question to answer. I don't have the answer.
Marie has another question. Marie, do you want to unmute yourself and ask the question?
Yes, great talk, Priscilla. So since there's all this topographical organization between DS and SN and SN and DS, I was wondering, do you think there's like not only one ascending spiral, but actually many that might, like be specific to specific movements like turning, licking, and so on?
Yeah, it's possible. Again, these one thing that I have not looked at, but has been suggested to me, is to look who are these SNR cells that are DMS targeted, and is this? It's probably not a homogeneous population, so it's very likely that you have all kinds of SNR cells here projecting to different places. So one experiment that I may or may not do, depending on time, is to check who are these. Yeah, I don't have the final answer because everything is so loopy. But you're absolutely right. I do think you can have these different loops for different things that are part of this bigger ascending spiral.
Great. Thank you so much. For the sake of time, we have to stop our session here. If there is remaining questions, I encourage the audience to get into contact directly with the speakers. I'd like to thank Marie, Claudio, and Priscilla again for their very nice talks. I really enjoyed them. Thank you so much. And also for the audience for participating and listening in. And I hope to see you guys soon again. Take care.
Thank you.